Hi, all. We're back. And I wanted to take an opportunity to chat with you a little bit about some things that are referred to in the textbook and a couple of things that are not referred to in the textbook. So, part of what I want to do is give you a concept of where genetics has been in the past, what has done to get us to the point that we are these days, and of course, where's it going? What are some of the problems that we can attack using what we know about genetics? And this really becomes the crux of my philosophy of the background of genetics and the importance of genetics in the future. Clearly, it's been influenced a lot by my international experiences, especially my time at the University of Cairo, spending several months in Egypt and in the developing world. And again, you have to remember that this was back in the 90s. Really changed my point of view about my career, the topics that I investigate, and how I approach them. So I hope that this gives you a little bit of that flavor. I want you to recall a little bit that some of the ideas that we talk about are really kind of old. And we really go back to the father of modern biology, Ernst Haeckel. And Haeckel was the first a person to actually use a tree, even though Darwin predates him by a couple of years and talking about making these little sketch drawings of how individuals can be related to one another and how species are interrelated. Haeckel was the first one to actually draw it on a tree. He was also the founder of what we called biogenetic law, which influenced uh, biology in the areas of biological study for decades. And the biogenetic law was termed ontogeny recapitulates phylogeny, and really flows off the tongue, makes you seem really, really smart because it has a lot of words in it that not a lot of people really understand. Haeckel was the one who invented these terms. Phylogeny, of course, very easy. We're talking about the relationships among and between different sets of organisms. Ontogeny is a really interesting one, another term invented by Haeckel. Ontogeny meaning development, and very specifically, Haeckel intended ontogeny to mean embryonic development. Recapitulates is a great word, and it just means to summarize, or this is actually where the word recap comes from. So when they say, let's take a look at the recap, well, what they really mean is they want to recapitulate what happened. Okay, so summarize, briefly summarize. But for Haeckel and for biologists, when we talk about ontogeny, what we're really talking about is embryonic development. So Haeckel felt that you could classify organisms based on their embryonic development. Turns out this was completely incorrect. However, and really influenced decades and decades of work trying to prove biogenetic law and giving us a point of view that was totally incorrect, and took us really well into the 80s before it was 1980s that it was generally accepted to be incorrect. I hope you all get a chance to read at some point in time a text by Stephen Jay Gould in which he really turns Haeckel's biogenetic law on its head. So I highly, highly recommend that as part of your reading and education in the life sciences. We owe Haeckel a lot. He talked about ecology. If you ever wonder where the term ecology came from, it came from Haeckel. And Haeckel, in addition to being the father of modern biology and a brilliant theorist, even though some of his theories were incorrect, was he was a tremendous artist. And you can take a peek at this one particular graphic that I really enjoy that was a picture of jellyfish. Okay, and so he had these tremendous drawings, and many of Haeckel's illustrations and depictions of living things are still used in modern life drawing classes today. So really, it's funny when you talk about Ernst Haeckel. The two classes of people who always know Haeckel are biologists and, of course, artists, because they tend to have seen a lot of his classic illustrations and/or drawings of things that he observed in nature. So tremendous artist. If we were to think about the background and the history, and we've already mentioned our buddy Darwin, you'll see this slide a couple of times during our lectures, and I like to start with this one. As we discussed previously in our first meeting, of course, we all know Darwin uh, for his text on evolution, and we credit him with that. But I really like the book that he published prior, which was The Domestication of Plants and Animals. And in it, again, I really look at the domestication of plants and animals. Even today, domesticated plants and animals are really the laboratory of evolution. That's really where we can watch evolution in our own time scale. And so we'll see some examples of this as we go along. Here, of course, is a great map. It's a horrible map of the world, in part because it is completely miscolored, mischaracterized. I think it comes from the late 1980s. And the only reason I keep it around is because I've been using it so long. I really don't like the color scheme. I don't like the fact that it misrepresents relative sizes of the different geographic areas of the world. Remember, all our geography, all our land masses exist on a globe, which means they can't effectively be flattened out in only 2 dimensions. This is a 3-dimensional structure. So it, by nature and by definition, tends to misrepresent the relative sizes and places of the different land masses. So that's something you need to keep in mind. And this is a great picture. I like it primarily because it shows you where the original wild ancestors were of the plants that we currently have and are domesticated. It is not an all-inclusive list, but it is a really nice broad spectrum across all of the different geographic areas in the world. Of course, when we think about our oldest plants, they're located right in the center in this, what we know as the Fertile Crescent. And this is where our oldest plants that have been domesticated, wheat, barley, onions, peas, lentils, chickpeas. A little bit later on, some of our fruits, figs, dates, pears, believe it or not, and really importantly, apple. And we'll see a video a little bit later on in the semester about the domestication of the apple, its origins and how it's grown and how it works today. There are a couple of readings that are included in Blackboard for you to read a little bit more about the apple. But these were all domesticated on the order of 50 to 75,000 years ago. So they're really quite a long time ago. And as we spread out from the Mesopotamian Valley and think of it as ripples from a pond, as human beings spread from their origins, we begin to see all of the different plants that were domesticated at a later date. Some of them are rather surprising when you think about the macadamia nut, for example. We always think of macadamia nuts as being the little bottles of Mauna Loa nuts that you bring from Hawaii, when in reality, the macadamia nut was originally domesticated in Australia. If you take a peek at North America, you see, of course, we mostly domesticated from North America fruits, plums, blueberries, cranberries, of course, big product in New Jersey, pecans, and sunflowers. And we'll see some other information about sunflowers a little bit later on. We tend to associate sunflowers with Van Gogh's paintings in the fields of France, but in reality, the sunflower came from North America, was brought back to Europe, and some of the old sunflowers were actually bred into some of the beautiful varieties that we have today. Uh, One of our most important plants, again coming from the New World, in the Americas, of course, is maize and corn. Maize, tomatoes, cotton, sweet potatoes, all very important food crops, all came from the New World. One of the fun ones that I like to point out is that yams were domesticated both in Africa and South America, and the feral ancestors, the wild ancestors, before they were altered for human purposes, can be found in both continents. So, kind of cool that we see this biological representation of tectonic drift, and that is that probably at one point in our past, the continents and the land masses were actually very, very close together and shared some of the same flora and fauna. And then as they drifted apart, and again, remember this is over a very, very big time scale. It didn't just happen in a couple of years or a couple of days. We see that human beings in both areas tend to take advantage of the local plants and animals and began to breed them for their own purposes. Of course, you have to look at the animals. When we look at the animals, again, we see here are some little graphics of all these different animal species. Again, not all inclusive, but we begin to see them again coming from the same basic area where humans began. Not surprising at all. Curiously, at the time I made this slide, I was unable to find a little tiny black and white icon of the oldest of the domesticated species. And probably many of you have that a representative of that species at home. Of course, I'm talking about our little puppies. Okay, dogs were originally domesticated, again in this general area. Same in the Fertile Crescent area, and dogs were domesticated probably from a hybrid of the red and the gray wolf. That appears to most contemporary or the well accepted version of domesticated history of the dog. Dog about 25,000 years ago. About 25,000 years ago. So the dog is actually the oldest of our domesticated species, and obviously from a single hybrid ancestor of a couple of different or closely related types of wolves. We have hundreds of species of dogs, and we'll see a little bit of a pedigree shortly. Next up, of course, came our buddies the sheep and oxen. These were, again, all domesticated in this same general area. So, but on the order of about 10 to 13,000 years ago. Again, nobody has a precise date. We didn't keep track of anything. We don't have breeding records from 10,000 years ago. And you can kind of see this. This is really a great little graphic. We can really chat about almost every species. Notice that unlike plants, there are no animals represented in the New World. That's not entirely true, however. There are a couple of species that were uniquely domesticated in the New World. I just didn't have little black and white icons of them. In North America, we see a species that eventually was actually considered to be the state bird of the United States, and that, of course, was the turkey. The turkey was domestic. The original turkey came from North America, and again, exported into Europe, where a lot of breeding was done, and a lot of the later domestication processes actually occurred in Europe in the neighborhood of about 1,000 years ago or so. South America, interestingly, another animal that was domesticated that many of you may have or have had at home, and that is, of course, the guinea pig. The guinea pig, our buddy, developed in South America and was developed not to be a fuzzy little pet that you can keep at home in a little cage, but rather was developed as a source of meat. It was developed as a food item. So when you go home and feed Fluffy, say goodnight, you might think about gearing up the barbecue. A lot of animals were domesticated for purposes that we don't really think about today. So a really interesting background. One organism that was domesticated that really gives us a tremendous amount of information about genetics, about the process of domestication, of course, is the laboratory rat and the laboratory mouse. The laboratory mouse is the most populous animal of our domesticated species. There are so many, used predominantly for biomedical research, was originally domesticated about 150 years ago in Paris, France. And we actually do have breeding records of both the laboratory rat and the laboratory mouse, both domesticated in the labs in Paris, France. And so we really know quite a bit, and they've contributed so much to our understanding of basic physiology and also basic genetics. More recently, we're seeing, as human beings gather control of the entire world and all the different ecologies represented on the globe, we begin to see domestication of other species. Shellfish, for example, are actually considered to be domesticated these days. Conch are being domesticated, mostly at Scripps Institute in California. So there's really quite a bit of interest in domestication of other species for food products to increase the amount of protein available for humans. So really fun. We could spend almost an hour or more talking about just this simple graphic here. I like to show you one of my favorite slides I just found a couple of years ago, and that is a slide showing the ancestral species of our citrus families. And really, all of the citrus that we eat today come from 5 different species. We've got the kumquat, the small flowered lime, the citron, the pomelo, and the mandarin. All are the old original ancestors of everything that we see today. And of course, you could see all of the different crosses. This is a wonderful representation. The very dark lines indicate the female parent. The light lines indicate the male parent. And any dotted lines, which are a little bit difficult to see here, but are available on the slide sets in a different folder, show you mutations that resulted into some unique fruits. You can kind of see how the pomelo, which looks very grapefruit-like, did result, in fact, in the production of the grapefruit. However, it occurred with a cross between some of the other populations here. So we see that the pomelo eventually became partly related to our contemporary oranges as well as grapefruits. Note the blood orange, that bright red. You may have seen it on the Food Network or any of the cooking shows. Actually was a mutation that arose from and was identified quickly from the orchards of sweet oranges. We see that the one of the really kind of curious ones is this thing called the Buddha's hand. And Buddha's hand almost looks like an inverted bunch of carrots, but is actually derived from the citron. It is not really used for food or juice, but is actually used in herbal medicine. And you can kind of see, if you do a quick Google search on those, they're really, really interesting looking beasties. And this is the Buddha's hand. So gives you some idea of the tremendous variation and all of the work that had to be done in both selection and also crossing of the ancestral species to create our contemporary array of citrus fruits. More recently, with whole genome sequencing of all of these organisms, we can actually put together a pedigree that is much like our previous graphic, which was based on archaeological and historical information, and we can now verify that by comparing genome sequences among and between all of the different fruits. And you can really get a sense, if you look at this kind of orangey, large number of orange bars in the bar graph, this is really showing you the importance of the mandarin ancestral variety to the production of all of these different fruits, including, of course, the kumquat. We see the different limes and lemons, and in fact, the contribution of the mandarin and its contribution, in addition to the pomelo, to create our contemporary sweet orange. So really kind of an interesting reflection of what we know from historical and archaeological information. We can verify that by investigating relationships among and between the different genomes and the genome sequences. So we'll talk a little bit more about how that's done toward the end of the semester. We can do the same thing, of course, looking at the ancestral crops. Here we see again the Fertile Crescent, and this is where all of our oldest of our domesticated plant species came from, also known as the cradle of agriculture. And so a very, very important part both for human evolution and, of course, what humans did with the plants and animals that existed around that time. And again, we see the founder crops, different varieties of wheat, barley, lentil, peas, and of course bitter vetch, which was not on the previous slide. To me, one of the most interesting aspects to come out of this, of course, is looking at a very important phenotypic characteristic for how do we determine which of the species gets domesticated. And when we think about plants, there's a really important characteristic of the seeding in plants that determines whether or not they can be domesticated. And this is a property of plants known as dehiscence. Dehiscence. And what dehiscence really is seed shattering. If you think about it, one of the most dehiscent plants we see in the Americas every spring and summer is our buddy the dandelion. And when the dandelion seeds mature and form that puffball of seeds, and you blow on them, of course, making a wish, you blow on them, the seeds disperse. And that dispersal of seeds, the breaking of the seed off from the main pedicel down at the bottom, what happens is it breaks off, and that is the process of dehiscence. And here is a picture right here, this cassulath grain in frame A. You can see the dehiscence of ancient rice varieties. That is, the seeds broke off from the main plant and were dispersed, whether by the wind or an animal came by or humans shook them off and made a wish. Now, can you imagine how difficult it would be to collect the seeds of rice if the seeds always broke off the stem? And of course, it would be nearly impossible. Imagine picking a dandelion flower and trying to collect every single seed from the dandelion flower. And of course, it would be very, very hard. But it's clear that in the very early days of domestication, and certainly probably during the days when humans were foraging, there was an identification of some types of rice that were indehiscent. That is, they did not shatter. And so, as a result of this lack of shattering, we were able, meaning humans, were able to collect the rice, use it as a foodstuff, and begin the breeding techniques. And as it turns out, interbreeding, making a series of F1 and F2 crosses between dehiscent and indehiscent varieties of rice, using restriction fragment length polymorphisms, which we will talk about a little bit later on, to map the genes within the F2 cross, we were able to identify a gene, a single gene on chromosome 1 in rice, QSH1, which was entirely responsible for rice becoming indehiscent. Really quite exciting. So here is a great example of a single gene mutation that must have occurred that humans were able to take advantage of and really allowed us to domesticate rice. So a very cool example of genetics influencing human behaviors and basically selecting the particular strain of rice that we were able then to collect seeds from and domesticate. The reason I find this really important in plants is several years after this work was done, another experiment was done with wheat. And guess what? Wheat, the wild ancestors, were in fact dehiscent. That is, the wheat granules fell off, broke off from the pedicel or the panicle of the plant and basically dispersed, just like our little dandelions. However, domestic wheat are completely indehiscent. That is, the seed stays attached to the plant until it's picked up by human beings. After this work was done, again, series of crosses were made among and between both dehiscent and indehiscent strains Again, genetic mapping, as it turns out, perhaps unsurprisingly to you, but surprising at the time, it turns out that the gene responsible for this change was a single gene in the homologous region of that QSH1 from rice. That same protein that was coded for by the gene has a mutation in it that causes the wheat to stay attached to the plant, again allowing human beings to collect the seed, use it as a foodstuff, and then eventually begin breeding the plant itself. So really an exciting idea that we can have a single gene influence the domestication of a trait. So we begin to see this. Just to give you a couple examples, one of my favorite examples, and one really is looking at brassica. It is the genus of the wild mustard plants. You've probably seen wild mustard. Wild mustard, you've probably seen it in your backyard. Wild mustard itself is a ubiquitous weed. It's a very, very small plant, just a few inches tall, maybe 4 to 6 inches if you let it to grow wild. It is a very, very small plant, has these lovely little clusters of yellow flowers, and of course produces tiny little seeds. And brassica, being ubiquitous, was used by human beings to create all of these domesticated varieties. So if we take a peek at some of the plants that were bred from the wild mustard, we see kohlrabi, kale, broccoli, Brussels sprouts. Love to see Brussels sprouts grow in the wild or in the grocery store. Cabbage, and of course cauliflower. All of these are related and can be tracked backwards to the original weed of brassica. So this is quite a fun one, and you could see different aspects of the plant, sometimes taken advantage of by individual mutations, just as we saw in rice and wheat, and other times just due to simple selection, that is, choosing the parents out of a wild field, choosing the parents that looked most or had the phenotypic characteristics that humans most wanted to use. So we have this kind of interesting array. For me, perhaps even more interesting, is taking a complete look at all of the different genomes that have been associated with brassica. And here is a great indication of the hundreds of varieties of vegetables that are all related back to brassica. Of course, you can see our cauliflowers and broccolis are very closely related. Brussels sprouts, kohlrabi, kale, and our more traditional cabbage that we make coleslaw from, all derived from the original brassica shown in the central portion here. And then take a peek at all of these other related plants that came from brassica, wild mustard. Many of them are Asian vegetables. You could see all of the different varieties of Chinese cabbage that have been derived from wild mustard, including bok choy, pak choy, but also turnips, parsnips, all of these different types that were also derived. So really tremendous power of human beings able to manipulate genomes through traditional breeding techniques into a huge variety of organisms that human beings eat all over the world. So a funny little weed that eventually really fed the world. Of course, the other weed that fed the world is, of course, maize. Maize is a very uh, ubiquitous plant across the world these days, and we'll be presenting this in laboratory when we talk about some of the early genetics of corn. And so I won't spend too much time talking about that, but I do want to point out one element of how the weed became a wonder. And by the way, that's a Easter egg or a little foreshadowing of the video Weed to Wonder, which talks about how we have contemporary corn plants that came from its ancestor teosinte from Mexico. And you can kind of see, but I want to talk a little bit more about what's happened more recently, and that is in the domesticated corn crops from the late 1800s to the early 1900s, yield in corn hardly changed. There was a nearly 100-year period where yield of corn was stuck at about 25 bushels per. I'm blocking here for a moment. This is about 25 bushels per acre. I don't think it's yes, bushels per acre. Thank you. But look at what happened in the 1940s. And when we look at the 1940s, it really became a period of time when breeders began intercrossing different varieties of the open pollinated variety and took advantage of hybrid vigor. Another term for hybrid vigor is heterosis. And so we begin to see the use of a double cross, and this is basically a four-way cross, which we'll talk about a little bit about in lab. And you can kind of see how all of a sudden, by taking advantage of a simple genetic idea of breeding hybrids, making crosses, you could see that the overall yield of corn tripled in the space of about 2 decades, about 20 years. Ultimately, the continued use of crossing throughout the 1980s and to today, actually, you can see that again, we see this incredible doubling of production to the point where it was not unusual in the early 2000s to see 120 to 150 bushels of corn per acre, and of course, the utilization of genetically modified traits, and in particular, the trait that was genetically modified was the inclusion of insect resistance, and we'll talk about that a little bit later on in the course and how that worked out. But notice that the slope of this line is fairly stable across the last 75 years or so, and we see continual improvement. To me, one of the most amazing things about this continual improvement is that we haven't run out of genetic variation. We are continuing to increase the yield of corn as time goes on in every generation that we breed corn. So year to year, a generation is about 1 year in corn. And so we see this continual increase thanks to genetic technology. So we're really quite amazing what humans have done to this point. I think this is a good stopping point for now. And so we'll take a minute. This will let you catch up a little bit. And we'll proceed talking a little bit more about the implications of all of this increase in production and the utilization of domestic plants and animals and how that's influenced human populations in the next mini lecture. Take care. Stay safe. See you next time.